Today is the last lecture of the semester for Stats 115 and 215 Bio Stats and 282. I want to first give a final review for the fourth module. We focus on cancer genomics and cancer immunology. The very first lecture is on mutation analysis based on whole exome sequencing or whole genome sequencing. The algorithm that was covered by Dr. Gadi Getz is MuTect, and it's still considered one of the best algorithms for mutation analysis. Usually for students in this course, you probably don't really need to start from the FASTQs. Instead, projects such as TCGA or these genome sequencing efforts, they will give you the mutation calls in a MAP file or a VCF file, and you can just take that for the downstream analysis. In order to really annotate the mutations, you can use algorithms such as Cravat to see, what's the consequence of those mutations. For MuTech group, they also look at recurrent mutations, which can be driver mutations by looking at mutation calls across many samples using the MUT6CV. And from there, based on the driver mutations, you can see cancer are caused by either gain of function mutations on the oncogenes or loss of function mutations on the tumor suppressors. And we showed you in the course some of the features of oncogenes versus tumor suppressors. What do their mutation patterns look like? What does their expression patterns look like? And what their copy number variation patterns look like? And sometimes you can also see cancer type specific mutation patterns or mutation load. And these cancer type specific mutation distribution or mutation patterns sometimes can be related to smoking, to some specific exposure to carcinogen, but also can be related to expression, to replication timing, or to chromatin states such as DNA methylation. And also finally, we talk about copy number variation analysis using GISTIC. So most of the time, oncogenes can also have copy number gain, and tumor suppressors can also have copy number loss. And so that's the kind of the whole mutation analysis. Most of the time, people do cancer subtype analysis, not from mutation, but based on the expression. Subtype analysis, people also use microRNA or protein expression, but most predominant approach is the expression approach. And so we talk about using gene expression, do clustering, look at subtypes, and there are also epimutations, which are subtype of the same cancer with different mutations on epigenetic regulators, and also on DNA methylation patterns. For example, the SIMP, which is a very special type of CPG island methylation phenotype on subtype of cancers, which is related to mutations or viral infection status and have implications of survival or treatment options. And once you have the expression pattern, you could also look at single sample GSEA or based on the whole cohort, look at co-expression network to see different gene modules. And the fact we are interested in the different subtype is because the different subtypes, for example, for breast cancer, whether it's luminal or HER2 or basal or triple negative breast cancer, they are treated with different drugs, and also the patient have different survival. So that leads us to survival analysis. And we know that different from other analysis for survival, there is censoring, which means patient could leave the monitoring. Either they stop getting the treatment or they passed away. And so how do you really look at the data? And for survival analysis, we look at log rank test or Cox pH regression. And so just be very careful when you try our survival analysis. When you have a very big gene list, by weighting the genes differently, you might always get some survival differences. The question is, if you have similar size gene list, you also get significance. So make sure you do some simulations to check that this gene list is indeed more important than other random gene lists of the same size. Once we look at the expression and echo exome sequencing, as we mentioned, the key is what do we treat the patient with? So we discussed targeted therapy. We started with early days when people were only treated with radiation therapy or chemotherapy, but targeted therapy is really targeting on the mutations, on the copy number changes, on the unusual high expressions, or on the fusion genes, right? So we need to look at what's the actual drug target. What is the mutation on that drug target gene? Does it really target specifically up or down regulated pathways? And in terms of drug resistance, do we see mutations that evades the drug from going into the enzymatic pocket? Or do we see parallel pathways or upstream, downstream pathways being activated to bypass the original drug target pathways? And also for targeted therapy, a very important mechanism of resistance is pre-existing resistance. And initially, they might only represent a tiny percentage of the population. But with the drug selection, it's being selected to become the dominant population. And in order to overcome drug resistance, we usually have to use combination therapy by looking at pathways that are synthetic lethal. So if you block one pathway, it's okay, the other is okay, but if you block both pathways, the cancer cell will die, but that will leave the normal cells intact. We also talk about chemical screens and genetic screens. So chemical screen is compound screen. You use one compound across many different cell lines, and you look at how much the cell is sensitive to the drug. And this is usually done looking at IC50 or area under the curve. And the cell lines that have specificity against the chemical or the drug will have a low area under the curve or low IC50. And we can look at those cell lines that are uniquely sensitive to the drug and see, do they share some expression patterns? Do they share some mutation patterns? And then we also talk about genetic screen. This is kind of the last part of the lecture, but I would also just kind of want to show here, for CRISPR screens, there are many, many different ways you can do the screen. You can use it to find essential genes, find drug targets, get biomarkers, understand drug resistance mechanism, find combinations, and we use MAGIC for CRISPR screen analysis, and also we discuss how to use depth map on the CCLE cancer cell lines. And then for cancer immunology and immunotherapy, we use two different lectures because there are many, many material involved. The first thing is, in order for the cancer cell to be detected as foreign, it has mutations, and those mutations are usually detected by us using genome sequencing or whole exome sequencing and mutation cost using MUTACT. But those mutations may not really be presented on the cell surface for our T cells or B cells to see it. And for antigen presentation, we talk about HLA cause. Depending on the HLA types of different individuals, different mutations will be presented on the cell surface. And so algorithms such as Polysolver can call the HLA type. And based on the HLA type and the mutations, algorithms such as NetMHC can predict which mutations will be presented by that HLA type to be presented on the cell surface. And so once the mutations are presented on the cell surface, you want immune cells to recognize those mutations. And the different immune cells includes T cells, B cells, and there are also class 2 presentation cells such as macrophage or dendritic cells. And so to understand different immune cell infiltration into the tumor, we want to show these immune deconvolution algorithms, which includes 6 different algorithms. And for TCGA, we use this timer website to pre-calculate immune cell deconvolution. So you can look at interesting associations. And then we talk about immune repertoires because it's the T cell receptor or the B cell receptor that really recognize the mutations. And so people have been doing TCR, BCR sequencing, and recently they're also looking at single cell TCR sequencing to look at what are the T cell expression status once the TCR is activated for clonal expansion. We can also use algorithm like TRUST to directly extract the TCR and BCR sequence from large tumor cohorts with just bulk RNA-seq. And because projects like TCGA have so many tumors, we can extract a lot of TCR and BCR and look at the association of immune repertoire abundance or constant regions in the antibodies and how they associate with mutations, with immune signaling, other immune cell infiltration, and so on. And then in order to really understand immune cell activation, people can look at the bulk RNA-seq. You can look at pathways using single sample GSEA. You can use immune cell infiltration estimation, but you can also use single cell RNA-seq or single cell attack-seq to see what is happening with T cells. What are the T cells going from, for example, naive T cell to a effector T cell, to a proliferative T cell, to an early exhaustion, to a late exhaustion in the tumor microenvironment, and how that influences the patient response to, or mouse tumor response to immunotherapy. We also discussed using ATAC-seq to look at whether there is the early T cell exhaustion epigenetic status versus the fixed T cell exhaustion status. So all of this, the RNA-seq and ATAC-seq are helping. And finally, the question is how to predict the immunotherapy response. And so we mentioned many factors that are important, such as mutation load. You want this tumor to look very foreign, and CD8 T cell infiltration. You want more T cells to infiltrate the tumor. You want T cell diversity so that different T cells can recognize different mutations. You want PD-L1 levels suggesting that the T cells there are already recognizing the tumor antigen starting the attack. So PD-L1 levels suggest that there are already some immune killing attempt at killing the tumors. Interferon signaling, you want to see whether there are bombs, being thrown at the tumor. And yeah, so there are also, computational algorithms such as TIDE to evaluate whether the T cells are in a really dysfunctional state or whether there are suppressive cells preventing T cell infiltration. Also, we want to use CRISPR screens to improve immunotherapy treatment. So kind of recap on this, using CRISPR, people can either edit the genome or have gene knockout, and also using CRISPR-A or CRISPR-I, you can have inhibition, also activation. And so related to that, in terms of CRISPR screens, we mentioned CRISPR knockout screens or CRISPR-A screens or CRISPR-I screens, and also Perturb-seq or Crop-seq, which integrate CRISPR screens with single-cell RNA-seq. And we talk about different computational approaches here. One is for designing CRISPR to make the cut efficient and not off-target. And so there are sequence preferences, and also try to target the functional domains of the gene, and also those conserved regions in the gene will give you a more efficient CRISPR guide design. And also for CRISPR screen analysis, in the homework, you guys are trying the MAGIC algorithm. For the applications, right, we mentioned here, we want to find the essential genes, the protein marker, how to use CRISPR screen to identify factors influence protein marker expression, such as MHC class 1 expression or PD-L1 expression on the cell surface. We use it to look at cancer vulnerability. We use it to look at drug response biomarkers and resistance mechanisms. And we mentioned this, in vitro CRISPR screen for cancer cells or immune cells, co-culture CRISPR screens of cancer cells with immune cells, or in vivo CRISPR screens where you put the cancer cells in the mice with different drug treatment to look at how cancer cells respond to different immune stimulation. And all of this help us in terms of identifying better combination therapies, combining different immunotherapies, combining targeted therapies with immunotherapies. And I think really to cure cancer, combination therapy and also early detection will be the way to go in the future. Okay, so that's kind of on the fourth module on cancer genomics, precision medicine, and cancer immunology. So I also want to give a final review for the whole course. I really want to congratulate the course students. You have learned so much thinking about when you started this semester, what are the things that we looked at in this course? There are many different biological questions we evaluated, right? So transcription, how to look at transcriptomics data, understand transcription regulation and how transcription factors are regulating the expression of genes. We looked at also epigenetics, different epigenetic marks, different epigenetic status, how chromatin factors influence the reading, writing, and erasing of the different histone marks and nucleosome positions, right? We look at how to use epigenetics to understand GWAS. We talk about GWAS, EQTL, and how to use functional genomics data to understand the GWAS hits. And then we talk about cancer genomics and precision medicine, targeted therapy, and cancer immunology, immunotherapy. So these are all really kind of wide array of biological questions. But you also learn a lot of genomic techniques in this course, right? So we started from RNA-seq, later on also single-cell RNA-seq. For transcription regulation, we look at ChIP-seq. For epigenetics, we talk about bisulfite sequencing, DNase or ATAC-seq sequencing, single-cell ATAC-seq, and also Hi-C for chromatin interactions. For GWAS, we talk about case control studies or family-based association studies, also EQTL studies. For genome sequencing, we talk about germline sequencing, which is mostly for GWAS related, and also somatic sequencing, which is usually tumor and blood comparisons to look at cancer mutations. For screening, we talk about genetic screens and chemical screens. We didn't specifically go over these, but you can see a lot of these genomic techniques are being combined. For example, CRISPR screens are followed with single-cell RNA-seq, or you see combined single-cell RNA-seq with single-cell ATAC-seq. And recently, there are also single-cell RNA-seq with proteomics. And also recently, there is also imaging or spatial transcriptomics. So you can really see genomics adding to other genomics or other omics assays together. And so I think things are getting more and more complicated, but also more and more powerful. So computational biology will become more and more important. We covered a number of statistics and computer science topics as well. You have to learn a lot of R, bioconductor, and Python programming in this course. We talk about quantile normalization, how to test whether something follow a normal distribution using QQ-plot. We talk about a lot of statistical tests, t-test, chi-square test, Kamenov-Smirnov test for gene set enrichment analysis, Mann-Whitney U-test for non-parametric test. This is for single-cell differential expression analysis. We talk about multiple hypothesis testing. Because in genomics, there are so many tests you have to do, we really need to care about correcting for multiple hypothesis testing, either using Bonferroni, which is very conservative, or false discovery rate, which is more popular in genomic assays. We talk about hierarchical models. This is kind of like RNA-seq. You are trying to borrow information from nearby genes to help you estimate parameters on an individual gene when you don't have so many replicates, right? We talk about generalized linear models, which is actually used in both LIMA for differential expression analysis or MAGIC for identifying differential guide abundance or differential gene selection in CRISPR screens. For clustering, we talk about k-means, hierarchical clustering, PCA, and later on in single cell, we also talk about using t-SNE and UMAP or graph-based network approach to kind of cluster the samples or visualize those cell populations or subpopulations, and also using the canonical correlation analysis to correct for batch effect. These are kind of the CS or machine learning approaches that people are using now in bioinformatics. We have discussed some machine learning approaches such as a support vector machine, random forest, neural network, lasso, and logistic regression approaches. And you've tried that in the homework as well. For sequence analysis for alignment, we talk about Boros-Waller alignment and also suffix array approaches for doing alignment. In motif analysis, we talk about expectation maximization and Gibbs sampler, and you can see how they kind of enrich on the different sequence patterns. We also talk about hidden Markov model and dynamic programming and how to use those to identify cell state, to identify TAD domains, CPG islands, and the copy number changes and things like that. And also we talk about survival analysis, right? And finally, we touched on many, many different bioinformatics algorithms. For next generation sequencing, we talk about FastQC and the BWA for read alignment. For RNA-seq, we talk about RNA-seq QC, which is kind of giving you the quality control, STAR for read mapping, SALMON for kind of pseudo alignment, and DE-seq for differential expression. For batch effect removal, you can either use Combat or Lima. And for gene ontology, we talk about David and gene set enrichment analysis. And for single cell, we mostly focus on Surratt, even though there are also many, many other analysis methods. For ChIP-seq, DNase or ATAC-seq, we talk about MAX and Cistron. And single cell ATAC-seq, you have some exposure on Maestro, followed by some Surratt functions. And for genome sequencing, I guess we didn't specifically tell you how to run MuTech because it's a pretty time-consuming effort, but at least you can look at the MuTech result. And we showed you how to do CRISPR screen analysis and also how to look at some immunology analysis methods. Oh, I forgot, actually, in this year, you also have homework really looking at how to do DNA methylation analysis and also Hi-C analysis. So I would say we have covered a lot of algorithms. And I hope through this, I know a lot of you guys worked really, really hard on the homework. And I hope you can see bioinformatics, we have no way of teaching you all the possible algorithms, but at least you can see you don't need to be afraid of learning another bioinformatics tool or looking at a new data set. You can have the confidence to say, okay, I'm just gonna read the README, try to install the software and try to start a run and evaluate the result and troubleshoot the results and understand what to do next. So that's what I'm hoping the course will teach the students. All these techniques are developed because there are new data types or new experimental techniques that have computational or statistical challenges. And so informatics algorithms are developed, some for QC, some for exploratory analysis, some for batch effect removal, some for comparison, some for integration, and some for kind of downstream more biological insights. But in general, I think through the course in all the homework, we try to make each homework as coherent as possible so you can see how people think about a biological question. Okay, So yeah, I really kind of want to congratulate the course on the students on working so hard this semester to learn so many contents, and hopefully, give you the confidence to go forward as the field continue to progress. We used to have final exam for the course, but because of COVID, everything is online. This year, we will not have a final exam. I want to still show you some things for final preparation. First, there is a lecture I want the course to watch. It's on the Canvas website. The lecture is about reproducible research, how to make the data and the code public so others can reproduce the result. But that video is really, when data is available, other scientists discovered that the analysis has been fabricated. And so we need to be very, very careful. Data fabrication is really a betrayal of your career. And also patients could be hurt. So we have to be very, very careful to make sure we're doing the right things, and by no means never fudge the data or the analysis. So I want every student to watch that lecture. It's really entertaining and also educational. And so this year we do not have the final exam, and grading will just be based on the 6 homework. And we would appreciate the students to fill out the course evaluation, and also even the last survey after you submit the homework 6. Please help us improve the course. We do try to take the students' feedback carefully and adjust the homework. If some questions are too hard and the students are complaining about some lectures, we do try to improve. And we try to make the homework better every year, and try to make it around the same hours of effort for each homework evenly, even though, I think every year we find some surprises here and there. So this year, some students complain that we have a lot of online pre-recorded lectures. I hope you could see that a lot of the pre-recorded lectures happen more earlier in the semester, because those contents have not really changed so much. These techniques are 10 years in the making, so we didn't really have to improve on the teaching too much. But a lot of later lectures, such as single-cell RNA-seq, single-cell ATAC-seq, cancer immunology, or CRISPR screens, these things are still changing so much. And so we really want to have live lectures so you can see the recent progress and the recent results in this field. So normally, if we have the final exam, we would ask students to answer different type of questions. So I want to just at least give students a taste of what the final exam look like. And so we can use this time to poll the students here. There will be usually some true or false questions. So let me ask, multidimensional scaling can perfectly capture the pairwise distance between nodes. Is it true or false? Can students enter your solutions or your answers in the chat window? Is it true or false? Can multi-dimensions scaling perfectly capture the pairwise distance between the nodes? The answer is false. Depending on your total dimensionality, MDS try to capture as much of the pairwise distance as possible. But if you only give it a single dimension, it will just have to do what it can. The more dimension you have, the better it captures the final dimensionality. But it by no way perfectly capture the pairwise distance. Second question, BAM file is usually used to store the coordinates of the genomic intervals. Is it true or false? The answer is actually false. BAM file is usually used to store the mapping of the reads. If you just want the coordinates of the intervals, you use a BED file. That's a much smaller file. BAM file usually is really big, and you just store the locations of the read. The third is epigenetic information is encoded in the DNA sequence. Okay, this one is easier. I guess the definition of epigenetics is the information, is inherited information not encoded in the DNA sequence. And the last one is a gene can be an oncogene in some cancers, but a tumor suppressor in other cancers. Is it true? Yeah, this is true. Yeah, many genes can be oncogenes in some cancers, but tumor suppressors in other cancers, sometimes depending on the mutation pattern. But those genes are, I guess, just really important in terms of gene regulation. They can't be too high, can't be too low. Either way would be bad. Okay, so next one. Usually in final exams, we also have multiple choice questions. So we want to ask, which of the following linkage consider pairwise distance of nodes in hierarchical clustering? Can people enter your solutions? Which of the following linkage considers pairwise distance of nodes in the hierarchical clustering? The answer is D, average linkage. Complete linkage is the maximum. Single linkage is the minimum. The pairwise, I guess, well, yeah, so average linkage is using the average pairwise distance. Single linkage is using the minimum distance of the, I guess, yeah, sorry, maybe this question is not as clear. A, C, D, there's no median linkage. The complete linkage, look at all the pairwise, but take the maximum. The single linkage is considering the pairwise distance, but using the minimum. Average is doing the average of all the pairwise distances. Okay, let's try another multiple choice question. Compared to SMART-seq, droplet-based single-cell RNA-seq method has lower dropout rate, lower profiling cost per cell, use UMI to separate transcript in different cells, and is more accurate transcript readout per cell. Which one is correct here? This is a multiple choice. There are multiple answers, multiple correct answers in here. Droplet-based single-cell RNA-seq is, it costs less per cell. The whole experiment may or may not be cheaper than the SMART-seq. But SMART-seq, the cost depends on how many cells you have to prepare. Per cell is more expensive. Droplet, the whole experiment cost about the same amount, but per cell is less. And droplet-based approach for RNA-seq, they do use UMI to separate the transcript in different cells. But SMART-seq has lower dropout, so is more accurate for in terms of per cell, the transcription expression level is more accurate in SMART-seq. Okay, so droplet is B and C, and SMART-seq is A and D. So next one is short answers. If you use hidden Markov model to predict the protein secondary structure from amino acid sequence, what are the parameters in this hidden Markov model, and what do they mean in this scenario? So what are the parameters in here? So hidden Markov model has initial probability. What is the initial probability in here? The initial probability in a hidden Markov model for protein structure prediction is, in general, if you look at protein structure, so protein structure prediction problem, the observations are amino acid. The hidden states are whether they are alpha helix, beta strand, or loops. And we're trying to predict the secondary structure based on the amino acid sequence. And the initial probability is, how often does the protein start with a loop or something, like so the starting point of the three structures. What are the transition probabilities? The transition probability is what is the likelihood that a alpha helix will follow the next, if one amino acid is an alpha helix, what is the chance the next amino acid again is an alpha helix, or beta strand to beta strand, loop to a loop, and also alpha helix to a loop, alpha helix to a beta strand, and so on, right? So that's your transition probability. What is the initial probability? The emission probability is, if you are alpha helix, what's the likelihood you are able to see amino acid, valine, glutamine, phenylalanine, and so on? If you are in the beta strand, what is the chance you are seeing a isoleucine or just different amino acid and so on? And so that's kind of how you use hidden Markov model to predict protein secondary structure prediction. So the next question is, if we have this table in here of comparing two groups, and we're doing hypothesis testing, right? And so what is the sensitivity? Can you write your answers directly into the chat window? If you have to write, x plus y divided by z or things like this, what is sensitivity? Yeah, this is the right answer. Sensitivity is if these two genes are if a gene is indeed differentially expressed, it is like or two groups different. Let's just think about the differential expression. Sensitivity is if the gene is indeed differentially expressed, what's the chance you're going to call it? And so if the gene is differentially expressed is M1, and you're able to call it is S, so S divided by M1. What's true negative rate? True negative rate. So these are the two groups are not differentially expressed, and you are not calling them. So it's U divided by M0, exactly. What is false discovery rate? What is false discovery rate? False discovery rate is among all the differential genes you called, which is R. What are the false positives, which is V? The V means the two groups are similar, but you call them different. So it's V divided by R. That's false discovery rate. Okay. What is a positive predicted value? Positive predicted value. Yeah, it's S divided R by R, exactly. So if you call it to be different, what is the probability that indeed they are different? So S divided by R. Okay, so I think we have lab notes on this. You can go back and check this. All right. And then there are usually some long answers. So throughout the semester, we taught you many different algorithms, and we want to make sure through these long answer questions that when you really deal with a biological data with some high throughput data, you understand how you string these different algorithms to analyze the data and generate hypotheses and get a paper published. And so this is really a way for you to see how these different methods are connected. So let's look at this scenario. In the tumor profiling study, your collaborator conducted RNA-seq of 50 breast tumors, and they're matched nearby normal tissues. And there are also some basic clinical information, such as patient age, tumor stage, survival, and there's no treatment. These patients haven't been to any treatment information. And so the preliminary analysis of the RNA-seq found that for many cases, samples from the same patients are clustered together instead of cancer versus normal clustering into two distinct clusters. So ideally, you want the cancer patients are together, all the normals to be together, or maybe the normals together, the cancers are at least in some subtypes. But in this case, you see a lot of same patients clustered together. Describe how you would like to analyze this data starting from FASTQ. And after the initial QC and normalization, are there any potential downstream analysis you could do to help your collaborator? Yes, so clearly this data, well, not clearly, but based on what we've seen, it seemed to have some batch effect, right? But since this is a long question, you say, okay, let me start from the beginning. We look at the data together. You can imagine you do some star alignment to map the reads, right? You do RCQC to make sure all the samples are correct. Like a QC is okay. You can use HT-seq. By the way, here there's no single correct answer. You can also imagine using Salmon to get to the expression index, right, to get the TPM. And then you can use either hierarchical clustering or PCA to cluster all the samples to really take a look, whether there are some outlier bad quality samples or whether there are some batches, right? And so you want to find out whether these are different batches in terms of processing, in terms of whether they're coming from different hospitals or things like that. You want to remove the batch effect from the RNA-seq. And then you cluster them again and see whether the batch effect has been removed. And after the batch effect removal, you might want to see whether there are subtypes in the tumor. So the ideal clustering is normal samples are together, and the tumor samples could be just one cluster or could be, 2, 3 different clusters. For example, if you were to compare the TCGA breast cancer, the normals should be together, but the cancers are divided into luminal, HER2, and the basal breast cancer. And then you want to calculate the differential expression between the tumor and normal, or also between different subtypes. You want to see, what's the difference between a HER2 versus luminal breast cancer. You see, luminal is high in ER and HER2 is high in HER2 expression. You can also look at their differential expression compared to the normal. And based on the differential expression, you can run either David or gene site enrichment, like a GSEA, to look at, gene ontology or pathways that are upregulated in cancer versus normal or in different subtypes. Because this is tumor sample, you can also kind of try other things. You want to see, do they have different immune infiltration? You can try immune decount, right? 6 different immune infiltration methods you ask. Does the tumor has more macrophage or more T cells? Do they show difference? And then finally, you want to ask, also you want to see maybe gender. Do men have more T cells than females, or things like that, right? You can also try things like TRUST to look at immune repertoires to see whether the immune repertoire is related to immune cell infiltration, is related to some signaling. You can try a single sample GSEA to look at the immune signatures in the single samples, or it doesn't have to be immune signature. It could be any signature you want to see, evaluate them. And also you want to look at Cox regression to see whether the tumor subtypes, immune infiltration, do they have, or gender, or, for lung cancer, you might also want to collect smoking status or age. Do you see difference of immune infiltration associating with age? Also tumor stage, and do they have effect on survival, adjusting for patient stage and tumor stage. Okay, so that's some ideas of how you would go about analyzing this data. Let's try another scenario. In the K562, this is a cancer cell line. The ENCODE consortium has generated ChIP-seq data over 100 different transcription factors and tens of different histomarks, as well as RNA-seq, bisulfite sequencing, DNAs, also ATAC-seq, and these have been pre-processed to pass QC. So you don't need to worry about QCing ENCODE data. These have all been QC'd. And so recently, a group discovered a brand new histomark. Let's just call them H2XYZ. I'm making it up. It's a new histomark, right? Oh, great, epigenetics. And they have generated antibodies, so now they can do the first H2XYZ ChIP-seq in K562. There is no perturbation. Let's just make it simple. Can you help her conduct the initial QC of this data to better understand what this new histomark, to just characterize this new histomark? What would you do? So you start from FASTQ file of this H2XYZ with some input control FASTQ files. What do you do first? So you can see here, this is similar to the other one. You start with QC, right? You want to do fast QC, you do the read mapping, you make sure it's not too redundant. You use max to call the peaks, and you also want to check the signal to noise in your ChIP-seq. You want to check the distribution of the peaks across the genome. You want to see, is my histomarks enriched in the promoters, exons, introns, or intergenic regions, right? Things like that. You might want to compare. You check the peak overlap with of the X2 XYZ with regulatory sequence, because there's like a union of DNAs. How good is that overlap? Does it happen in the enhancer regions, right? You can look at evolutionary conservation, and also you compare with other ChIP-seq using Systrom Toolkit to ask what other ChIP-seq has overlap with my ChIP-seq of interest. Does XYZ look like K27 acetylation? Does it look like DNA methylation? Does it look like estrogen receptor binding? Or does it look like some other, published ChIP-seq data? And also you can use, you can assign the reads of the XYZ to the nearby genes, and you can look at the putative target genes of XYZ. And if you have differential expression, if you have gene expression, this is similar to, remember your homework 4, when you were trying to look at DNA methylation. You want to see whether DNA methylation is associated with active gene expression or silent gene expression. You want to see whether the mark is appearing in the genes that are silent or active in the cell. And so at least I think that will give you some idea. So in this compare, actually, we're already using some external data in here, but you can imagine you integrate the ChIP-seq with the expression data with all the other external data out there. Okay, so that's roughly how you would probably kind of characterize a new histomark. All right, let's look at another scenario. So there are 12 different cell lines of cancer type X with mutation on gene A and 6 without mutation on gene A. So this is all of the same cancer type, but 6 with a mutation and 6 on wild type of this gene. And so you have generated CRISPR screen and RNA-seq and ATAC-seq of these 12 cell lines. How do you analyze that data? I would say it's always the same. You start with QC and process each data type. CRISPR screens by itself, RNA-seq by itself, ATAC-seq by itself, right? So initially, you do your QC, you do your batch effect removal to make sure the data is usable. The second is to compare the wild type versus mutant cell lines. In terms of RNA-seq, are there differential expression? In terms of ATAC-seq, you compare the wild type with the mutation. The CRISPR screens, you compare the wild type with the mutation. So second is usually compare. The third is integrate. How do I combine the different modalities? How do I integrate the RNA-seq and ATAC-seq of the same cell line? For example, through Cystron Go, you ask, are the genes that have more ATAC-seq peaks and also higher RNA-seq, are they more essential in the CRISPR screens, right? So you start to kind of look at your data that's kind of more integrated. And finally is to check the external data. If there are some genes that become more essential, if there are some pathways that are becoming more essential, you look at, say, TCGA, other public data to see what is the disease or clinical relevance of what you are finding in this 12 different cell lines, right? And so the last scenario I want you to kind of go back and think again, same thing. So we have 10 patients with immunotherapy, 5 responders, 5 non-responders, and you have generated single cell RNA-seq of pre and post-treatment tumors. So 10 patients each before and after immunotherapy treatment. And so again, you first want to do your standard QC, and then you want to do the comparison before, after treatment, responder, non-responders. You want to look at their immune cell status, immune cell infiltration level, differential expression, and things like that, right? And then, but by the way, once you do differential expression of the different cell types, you can do gene ontology, pathway enrichment, and the third is to check other cohorts, check immunotherapy, see whether other immunotherapy, to see whatever you are seeing is reproducible in, or at least it's a phenomenon holds in other cohorts. Okay, so that's kind of what the final exam should look like. Hopefully this will kind of help you bring all the first 6 homework together. Okay, I think, sorry. In the very first lecture for the introduction of bioinformatics and computational biology, we mentioned different levels of bioinformatics research. I want to just reiterate this so that you can, now that you have been in this course for the whole semester, hopefully understand the difference of the different levels. Level 0 is modeling for modeling's sake. We actually didn't bother to even teach you this. It's usually a question, a bioinformatics question that nobody really care about. They are not really published in high-profile journals. They are not really read or cited well. Hopefully, you don't even need to go there. And what we start in this course is to teach you how to do level 1 bioinformatics research. You analyze collaborators' data, or just basically you use public tools to analyze data and generate some hypothesis. You say, oh, you do QC, does the experiment work? Do we see some meaningful differential comparisons? Does that mean something? Is this related to the disease, right? Is it important biological problem? Are there existing algorithms to analyze the data? So if, for example, that lung answer I showed you is typical level 1 bioinformatics. You have some data, what are you going to do? Right? So you think, oh, what tool did I learn in this course? And check out, right? Or you just Google on the web, what algorithm can I use to analyze this new type data, experimental type, and try to learn this. But there are different levels of there are different, you can do the level 1 bioinformatics analysis well, which means that you understand what your experimental collaborator is trying to answer. You picked really interesting questions. You are using the algorithm in a kind of smart way. You help them generate the hypothesis. You even help them figure out the experimental validation to do in order to validate your hypothesis, right? And so that new experimental validation generate new data to help you refine your hypothesis and so on. And how much are you using public data to help them narrow down the hypothesis? Does it really have novel biological finding, or your collaborator already had a very clear idea and they just want you to help them generate the figures so that they can publish the hypothesis, even though you find the hypothesis to be wrong. You can see this, does the experimental figures and the computational figures alternate? That actually is a very important idea to see how much is the informatics analysis helping to refine the hypothesis, to help with the experimental validation, and then generate new data for the experiments, for the computational analysis. And so this is kind of a more iterative process. And also, usually if you see the alternating experimental computational figures, and usually the first and the corresponding authors will also be joined between the experimental and computational groups. So the familiarity of bioinformatics algorithm and understanding of the biological questions are very, very important. And through these collaborations, hopefully you can learn the domain biological knowledge as well, and motivate the methodology development. And just be careful. Don't be a data janitor. You want to look for collaborators who care about working with you to generate a hypothesis. They value your input. And if the data prove the hypothesis to be wrong, they are open-minded enough to even consider this new hypothesis rather than, here's our hypothesis, here's the data, I want to just generate this figure, please help us generate the figure, that's it. Level 1 bioinformatics is kind of what we teach students in this course to do. For level 2 bioinformatics is really for algorithm development, in order for us to QC, analyze the data, compare. So basically the algorithms that we teach you, those developers are doing the level 2 bioinformatics. I mean, think about BWA, think about Salmon, think about DESeq, right? The groups that develop gene ontology, GSEA analysis, Surat developers, and so on. So they have a keen eye on the most cost-effective new techniques and what are the questions, what are the remaining quantitative or remaining computational bottleneck, and they use sound statistics or best machine learning practices to develop a very good user-friendly software so that more people can utilize the result. So if you think about level 1 bioinformatics, it's not scalable. For every collaborator, you have to kind of repeat this analysis to regenerate different hypotheses. You can only use published tools to help one group at a time. But with level 2 bioinformatics, because they generate an algorithm, BWA is being cited by probably 20,000 times. Many groups can benefit from the result of this one algorithm, right? So is database and web servers. Many groups can use it. The key is for the developer. By the way, these are not necessarily published in high-profile journals. Of course, if you can publish in Nature Science, that's great. But a lot of the algorithms are probably just published in bioinformatics or genome biology or even lower journals. But if it's really used and cited a lot, that's the real value. And so it requires the group to continue to maintain and update the algorithm or the web server with new data and new, with new data or new challenges, providing new features to overcome those challenges and adding more features. And so in order for the group, the informatics group to do well, they need to understand their biological domain. For example, there are groups that are really good with alignment. They stay close to their alignment area. There are people who really care about epigenetics. They develop the algorithm focusing on transcription epigenetic gene regulation. There are groups that just focus on proteomics. There are groups who are just focused on small RNA or immunology or single cell. These are kind of very different domains. And in order to do well, you need to provide the complete solution. If you only just provide a little bit, you think about Surat now, a lot of these packages now provide from the initial mapping to the QC, batch effect removal, differential analysis, identify markers, visualization, downstream integration, everything together. And so if you care about that experimental technique or that domain, a lot of the solutions that become popular provide the complete solution to the whole domain's problem. As we mentioned, the user adoption is more important than publication journals impact factor. Even if it's published in bioinformatics, if it's cited 20,000 times, that's a really successful algorithm. So time is the ultimate measure of success. Is the algorithm widely used and widely cited? Do people use it to really make real findings? That's the key of a success. And here, the key is to do well by doing good. When you develop a tool that benefits so many people, so many other users, usually this person is getting the respect from the field and also getting more research funding to support their methods development. And then level 3 bioinformatics is now computational biologists in a biologist's role make biological discoveries, usually from public data integration and modeling. This is true data mining. Because there are so many papers already published with public data, can you really make a discovery? And so this level 3 bioinformatics research required the computational biologists to have their unique biological angle. They care about a specific biological problem. The algorithm can be developed in their group, but it could just be a smart use of existing tools, but answer a very interesting biological question or looking at the data in a fresh angle. So you need to make real biological discoveries rather than putting numbers on theories. So there are some previous theories already, and they are just trying to fake the analysis to support that. Hopefully it's a really novel discovery. One difficult thing is that sometimes these kind of hypothesis generation requires some experimental validation. And that experimental validation can be the informatics group themselves, because sometimes the groups that really care about the biological questions, they have some experimental capability to validate the hypothesis, but it could also be an experimental collaborator. So for example, my group engaged in the level 1 informatics to help my immunologist collaborator. And so hopefully if I generate a hypothesis, my immunologist experimental collaborator is willing to help me do some validation. Right? So you help them with their analysis, they help you with your experimental validation. And then recent years, there are more and more also level X bioinformatic research, which is integrative paper from big science consortium. These are team science, major consortiums. Sometimes you can see this ENCODE, TCGA, the whole like GTEx consortium and so on. So some of these papers have numbers on the paper title, like profiling of 1,000 lung cancer, for example. That you can see that's that's a level X team science project. So there are usually significant efforts used to generate resource for the community. Usually these level X papers, the data is more important. Yes, because they have so many so much new data, there is some interesting biological analysis. So the informatician helped to integrate this together to make some initial attempt at a discovery. The publications are usually in high-profile journals and highly cited, not necessarily from the finding, but even the data itself is worth a lot of citations. Although the authorship, who is on the first or last authors, sometimes depending on the internal politics of the consortium dynamics. And so the trainees who are in these type of level X team science papers, if they are the first authors, they usually get into good, they get a good first author paper in Nature or Science, they can get a faculty positions, but they need, they learn valuable experience working in a team, but they need to learn to become truly independent scientists. Once they become independent, like they're in a faculty position, they are out of the consortium. Can they stand on their own? Informatics groups should, I think a good informatics group should do level 1, 2, 3, and X. They should do all three, maybe with a slight preference for one, but try not to just do one alone. And also, especially the level X researchers need to be careful they don't lose the first three. Okay, so for advices to excel in computational biology for students in this course, if you want to be a good computational biologist, I hope you can love both the biological discovery and the quantitative skills, right? Try to actively follow the field from the literature, from going to conferences or listening to talks, from following social networks such as Twitter and blogs, so you know the state of the art. Don't be afraid to learn a new experimental technique or new computational methods. I'm hoping through this course, if you can, follow up to this point, you are no problem. You won't be afraid to really try it. For computational biologists, we have to be careful to bet on the new experimental technologies. It takes tremendous effort to develop a computational tool for a method. But if this is not so popular, you bet on the wrong horse, your method will not be cited a lot. And so make sure you focus on the technique that has more promise, because there, your method might be used by a lot of people, and also there will be more data generated on that data platform, which you can also later on do integration to make discoveries, right? And also learn to think of creative ways to squeeze more insights from public data. For example, ENCODE data or TCGA. I mean, the data has been generated and the original paper were published, but still more people are using the published data to make new discoveries from those. That's when a computational biologist really become the level 3. They can stand on their own, they can make discoveries. And also look deeper into unexpected results or outliers. A lot of times you might see, this result is kind of different from what you expected. Then try to use other public data to see, is this phenomenon just from my data, or it's actually a general phenomenon? And is this coming from the experimental technique problem bias, or this is actually a real biological phenomenon? For example, super enhancers. It's just, if you look at K27 acetylation signals, they are outlier genes. They're super strong, right? They have really wide and tall peaks. Oh, turns out these genes are super important. And so pay attention to those unexpected or outlier results. And because computational biology is interdisciplinary between statistics, computer science, biology, and genomics, having communication skills will make sure that we are able to talk to the different people with different domain knowledge and make them appreciate what we are doing, right? So you can really reach both the experimental biologists and experimental biologists too. You can talk their language so that they appreciate what you are doing. In closing, I want to show you, this is Susan Hockfield. He was the MIT president at the time, and in the World Economic Forum, she says, 21st century will be defined by biology. And I'm hoping that from the recent COVID pandemic, you can see how important biology is defining our lives now. I also want to show you, we have this in the first lecture, 2002 Nobel laureate in a 2008 interview says that we presently has huge amount of data that are largely unassimilated. I've discovered that while there are rewards of collecting data and distributing data, there's little, if any, efforts for organizing this. It is what I call low input, meaning not thinking carefully before, high throughput, meaning that you generate a lot of genomics, high throughput data, and no output science, meaning that data is not actually used to answer a real biological question, to make a real discovery. But this is saying that, well, first of all, 21st century is defined by biology, and biology is defined by high throughput technologies, but high throughput biology is defined by computational biology. If you can't really understand the data or analyze or making sense out of the data, you are just wasting money. And so I hope in this course, you have learned all of the useful things. And I hope you can take the skills now and practice a lot more computational biology and use it to advance biological discovery. Okay, so thank you so much for staying on in the course. I hope you enjoy the end of the semester and use informatics well later in your career. All right, thank you.